Does that mean we're starting, Jeff?
Yeah, you're good to go.
Okay. Hello, come on in. Okay, I'm going to start just a little bit early. I have like a minute before I officially have to start, but I'm hoping if you have any questions, you can ask today. You can also see me next week during office hours if you need to drop in. As I mentioned to you on Tuesday, if you study just what I'm going to go over with you today, you should be able to get like a 90% if you know your material. If you don't know your material, you should definitely come and see me. I want you to really do well on this, but I also want you to take away from all of this like the core material, which is really how do we do molecular, cellular, genetic techniques in neuroscience. And I want you to be able to feel confident at the end of it that you actually know that you're capable of understanding a paper, that you're able to read a paper and understand it, whatever the topic is. So I'm going to try to make sure that you're ready. I'm going to give you a really quick overview of the midterm. All of this is going to be available for you, so if you want to jot down a couple of notes, please do. Again, the video itself hopefully will go through without any problems, Jeff, hopefully. And then this entire package will be available for you as well. So the midterm itself is going to be 90 minutes long. It's not going to be written here. It's going to be written elsewhere. I'll be posting the location for you during reading week. The material only covers weeks 1 to 4, not what we've been doing this week. You'll have 25 multiple choice test questions, 10 application type, and I'll give you an example of it. It's really not as difficult as other multiple choice test questions you've written. What later on will involve labeling some diagrams, and I'll give you some examples of this, or drawing diagrams, not just labeling. Very short definition type questions, and part of that is we take aspects of what we cover here. So I want you to review it, to know it, and then when you leave after the test as well, to be able to remember all of these key points. Bless you, Elise. We have one short answer. It's not worth a lot of marks, but one short answer that's more application based. And I'll give you some examples of the types of questions. Then one of the questions actually you'll get exactly the way that it's written on your term as well, because I want you to be able to think about it. So this page, and you'll see that the multiple choice, you get 1 mark each, 25, and then you'll have 3 short answers with parts, worth 20, and they're really straightforward. It's kind of like in terms of neuroscience, it's like an action potential. Some are all or none, you either get it or you don't, and some are sort of graded responses. So you'll have to think a little bit about it. There's probably like 1 or 2 ways to answer the question, but not a lot of choice. Okay, so it'll be really straightforward. I'm imagining that most of you will be able to walk out of there feeling really good after about 60 minutes or so, but we want to give you the 90 minutes just in case. Do you have any questions about the format before I go on? No? So my hope is that during the reading week break, which starts next week, how many of you have already started studying for HMB360? So that's pretty good. How many of you intend to start studying next week, which should be everyone? You to take a look at. Feel free to discuss them on whatever form you want. I will provide answers for them the day before the test, so you can go back and check like what your responses are. But I want you to have a good feeling walking into the midterm. So any questions about the format, what you're looking forward to, etc.? You're not looking forward to it. I kind of am. I'm hoping you're looking forward to it. Okay, so I'm going to start off by what you need to know like in week 1 from lecture 1. So most of lecture 1 was an overview lecture. I don't want you to start memorizing everything. So for example, here is slide 20 from lecture 1 from week 1. I would not ever try to ask you to label this diagram on the right because I never went over this diagram for you on the right in any detail. So I don't think that that would be a fair question. However, I would ask you to, because we discussed it in class, be able to at least know some of the details, because we talked about a nonsense mutation and how it generated a stop codon, and that's in your notes a little bit later on. You don't have to remember all the details of things that I did not cover in class. So I will ask you to know details about the various forms of muscular dystrophy. And we talked about several different types, like Beck's muscular dystrophy, Duchenne's muscular dystrophy, and that Duchenne's muscular dystrophy itself is usually X-linked. Okay? So those types of details I would expect you to know. And memorizing things like this on the right would not be things that I would expect you to be able to label on a diagram because I didn't really cover it in a lot of detail. And this is actually more or less a summary of what we did cover related to Duchenne's muscular dystrophy. So I will tell you ahead of time that you should expect things in your multiple choice related to like Gower's sign. Do any of you remember Gower's sign?
It's kind of one of the first markers of muscular dystrophy when you have a hard time getting up. So you like children have to walk up like your legs.
Right, so they walk themselves up because of muscle weakness. And it's something that's common. It's not only related to humans. In fact, we'll talk about this a little bit later on, but it's one of those characteristic signs and features. If you see Gower's sign, really we're talking about Duchenne's muscular dystrophy. Of Duchenne 23, multiple choice. This is another example of a diagram that I did not cover with you in any great detail, so I wouldn't expect, like, don't go nuts trying to memorize this, that, oh, I'm going to have to label a diagram. This isn't one of those types of diagrams. You don't need to know anything from this particular diagram here at all, period. The one thing I'll take you five. I didn't really cover all of the molecular binding partners for dystrophin, which is this large molecule here. I mentioned to you that dystrophin is a large molecule that has many different components that also binds to other components that are found in the sarcolemma, which is part of the muscle membrane. But I told you that you don't have to know all of the sort of ancillary proteins that are found in this complex, and that's true. You don't have to memorize this. It's not going to be a labeling diagram where what am I pointing to here or anything else, okay? So I want to be very clear. What I cover in lecture, what I mention in lecture is what I expect. I'm not trying to get you to lose marks because you didn't we didn't cover it in detail, and you shouldn't be spending a lot of time memorizing this. It's really the bullet points and what I mentioned to you in class that are more important. So the other thing I'll mention on in terms, well, there is a deletion, but we still have dystrophin. Here in Duchenne's muscular dystrophy, the deletion itself is relatively small. However, what happens is because it causes a stop codon to be introduced, we don't really form dystrophin as easily. And so we know that this is actually the more severe form. So this diagram here, in terms of knowing that the size of the deletion is bigger or smaller, that's something that I would want you to take away from this slide. Okay, not exons are involved in this, how many exons are involved, but the relative size of the deletion. Any questions on this before going on? Okay, so in terms of diagrams, and this is slide 27 from week 1, lecture 1, I would expect you to be able to summarize this diagram or give me the major points. What might be different if dystrophin is lacking? What sorts of things might you expect to see? Because we did go over this diagram in terms of the types of positive feedback loops that are introduced when dystrophin itself, we have a stop codon being introduced in the muscle cells. What sorts of things occur without dystrophin being there, without that complex being anchored? What sorts of things are the downstream consequences? Example of a slide. Those labeling diagrams, I really wouldn't, because it doesn't photocopy well, to be honest. Like, would I normally ask it? I probably would, but when we tried photocopying this and trying to give it to you as a figure, it didn't really work well. You had a question at the back? Yep. Right, so activation of proteases like calpain, intracellular calcium becomes unregulated, the sarcoplasmic reticulum here on the right itself becomes another source of calcium, so it's a positive feedback loop. And in addition to that, it will also start the NF-kappa B cascade that leads to an inflammatory response. Okay. So other things that you should make sure that you know, again, think in terms of short answers, things that are yes, no, easily marked, where it's not, you know, too subjective or anything, like really objective answers. You should know some of the animal models that are associated with Duchenne's muscular dystrophy. In almost every instance of a genetic disorder that we went through, we talked about an animal model. And you should be able to give examples of an animal model for all of the different situations that we went through. One of those models is the MDX model. There are some advantages to the MDX model. There are some disadvantages to the MDX model. You should know those, and you should be able to clearly outline, like if you're asked on a short answer question, what are the advantages or disadvantages model. Okay, and let's just try one type of multiple choice test question. And again, you don't have to, if you want to and you feel like it makes you feel better, go ahead and take a picture, write it down, whatever you want. But it might sound really easy for you right now, but you also have to keep in mind that we're going through this lecture. You may not get it in the order that you're studying. And so here is the type of scenario, the type of question that you'll see in terms of application type. So Ron has found a gene that, when overexpressed, will reverse Gower-like signs or symptoms in golden retriever dogs. So do golden retrievers, by the way, are they a model for muscular dystrophy? Yes. Okay. So we know that some dogs, including the golden retriever, or muscular dystrophy, Gower-like signs, immediately tell you both of these things are telling you right away we're dealing with muscular dystrophy, right? So you should be reading these questions, the application types, specifically to that you probably want to look at. So this is one where I just wanted to know if you've actually started studying. So which of the following genes, in other words, would he insert into the AAV system to reverse the Gower-like symptoms? So Gower-like is found in golden retriever dogs. Information for answers, but some of you might also be thinking there's more than one right answer. So what do you guys think is the right answer here? Yep.
I'm thinking he could do a couple things.
He could do a couple of things. Actually, there's more than one theoretically right answer here, but you're given information enough to be able to answer this in a particular case. So think very carefully about this. Sorry?
I don't know. I think it's A.
You think it's A. Okay. Um, yes.
Question actually.
Yep. Yep.
Would Utrophin even work in a golden retriever model because it's a mouse?
Okay, that's a good question. That's a good question. Um, so this gets back into, we didn't talk about this in this review session. It's on some of the slides that I've just mentioned to you. What is Utrophin? Okay.
And it's sort of functions like dystrophin. We know that in the MDX.
Okay. So it's sort of like this. Okay. So that's one thing that should be drawing your attention. It's kind of like dystrophin. This is a shorter version of it. In many cases, it can take the place of dystrophin. How about NF-kappa B? If we overexpress NF-kappa B, does that do us any good? Probably not. It's involved in that inflammatory response. Adelorin. So that's a compound that we have actually talked about. Adelorin. Yep. So it's a drug that causes exon skipping that's used in things like maybe muscular dystrophy. But do we have a gene for it? We don't. It's a chemical compound, as I mentioned to you. And so adelorin in this case wouldn't be a really good answer to a particular gene. And CRISPR-Cas answer. Okay, so answer A is the correct answer because it's a gene that if we overexpress it based on all the other things that you know as an application type question, it would probably take the place of dystrophin. We did not learn about it in the case of golden retrievers. You're absolutely right. But going through this whole process, the best answer here is actually utrophin. And you should expect to see a very, very similar type question related to utrophin on your midterm, okay? See a lot of, are you guys okay? Yes? All right. Did you guys find that a really hard question? So these are like of the 10 application type, this is what I mean by application. The other ones are really literally straightforward. Like you'll know right off the top that the answer is this, okay? Any other questions before we go on? Jeff, do you know if this is actually recording? You don't. Okay, we'll just.
I mean, it's probably recording.
Okay, hopefully. Okay, we'll hope for the best.
Let me check.
That's okay. We'll so slides from tutorial 1. So you're kind of wondering, what about all the 10? The stuff in between slides 10 and 13, you probably want to skim over, but I would start at slide 10, forget all the other stuff. I'm not going to test you on it. Just get started on reviewing different genetic techniques. Some of you will know the Krelok system. Some of you may have heard about TALEN systems for the very first time. And then, of course, we spent a lot of time on this CRISPR-Cas9. I am going to ask you to make sure that you know the details related to the CRISPR-Cas9 system as is pointed out to you in the bullet points. And the questions will be taken directly from the bullet points that are on those slides. I think that going forward, all of you realize that CRISPR-Cas9 is going to be an important technology. And I want you, and I know you're not going to remember everything about it, but I want you to remember a lot about the technology kind of moving forward. So I do expect you to know the summary slides that are associated with this technology here, as well as the CRISPR-Cas9 system. Okay, so the other thing that you should be able to do is to summarize the advantages as well as the disadvantages of the CRISPR system versus RNAi, for example, or the TALEN system, for example. You should also be able to tell me, like, what are some of the technical considerations? Well, one is you need a PAM sequence on the alternate strand. That has to be in that DNA that you're trying to cut. So that's a restriction or a technical consideration. You need a certain amount of GC-rich content within the primers that you're developing. And so on slide 20 of this tutorial, you should be able to summarize, because they're important points. When you read papers now, I'm hoping you're not just sort of bypassing them. So you're going to see a few multiple choice test questions on this section, so I won't lie to you. This is a small tutorial, but it is a tutorial where I've written some multiple choice test questions for you. And you're also likely to see short answer questions as well as potential diagrams related to this, okay? Again, really easy diagrams if you know them, and you're able to go back and review diagrams like this. You won't have any difficulty at all in this, okay? Any questions before we go on? So let's so we want to know if someone's been using a genomic editing technique on these mandrake plants, causing them to die. The one thing, and again, this is not even an application question. This is like, if you know it, you know it. If you don't, you won't, okay? Is if you studied. If talens, using talens, be like the again, what would be left over in the TALEN system? Does the TALEN system care if there are PAM sequences or not? Does it care if you have guide sequences? Does it use a zinc finger nuclease? Does it use FOC1 enzymes? What is characteristic of TALENs? Yep. They use the FOC1 enzyme, and that's the type of example where either you know it or you don't. And about 15 of those questions are literally, have you studied your notes? That's it. I'm not making it tricky at all. I just want to know if you have studied your material, because it's a basic technique, TALENs. It uses the FOC1 enzyme. It's different. They don't have guide RNAs. They don't have PAM sequences. And as long as you've studied your notes, I'm hoping it's really straightforward. Does it seem straightforward? Yeah. And again, you don't have a lot of lecture notes to go through. Those are important techniques, but I want you to spend time going through them. So I know for the genetics students, a lot of you are a little bit concerned about some of the electrophys and everything else. For week 2, lecture 2, when you start going through it, you do not have to know what an EPSP is, unfortunately. For your own information, I'm hoping that you'll take some stuff away from you with you in this class at the end of it. You don't have to know slide 7, which was that sort of diagram about whole cell recording and about inside out and outside out patches. Again, as much as it kills me as a neurobiologist, this is a neurogenomics course, so you don't have to know all of those. But I would actually ask you, this is slide 10, and on slide 10, like, you don't have to worry about all this stuff over here. In fact, what we do in the subsequent slides over here is we go through the NMDA receptor, and we go through the AMPA receptor, and we talk about the subunits. And that is something that I want you to know. And the information on slide 10 is really the bullet points that follow on slide 11, slide 12, etc. Any questions about this? So basically, from slide 10, you should really know that the NMDA, AMPA, and kainate receptors are multimeric subunit glutamate responsive channels, and that's really it. Okay. Yes.
Did you say we had to know their segments?
You have to know that they have subunits, yes. And on the subsequent bullet points that follow, we actually go through the AMPA receptor subunits, that they're heterotetramers, that they have like 4 subunits, and that we have like 3 different types of glutamate responsive ones, NMDA, AMPA, as well as kainate. Doable? Of course it's doable. And this gets into slide 13, which actually addresses what I was just mentioning to you. So this becomes one of the biggest things that I want you to take away from all of the slides, whether you are aware of it or not. Slide 15 is actually an important slide in a couple of ways. And I want you to think about this really carefully. How would you know if you actually had performed CRISPR and it worked? How would you know? So one is they do electrophysiology, they go back and check for NMDA. That's one way. How else would you be able to tell? Yep.
Can you use a Sanger sequence to see if you've deleted that sequence?
Okay, so that would give you, if it was at a 2, sequencing, I don't really care how you do it. Sanger sequencing is the old way to do it.
Yeah.
You could use the next generation platform. That's okay. Is it only deletions that cause disruptions? Is CRISPR only going to delete?
No, they can also do insertion.
Right, so if you had given me insertions and deletions, or even if you used the short form indels, I would be okay with that. You would have gotten full marks. If you had only put deletions, I wouldn't be sure if you really understood how CRISPR works. It's breaking up the reading frame, basically. And this is on the slide several times. So how would you know? You would actually have to go in and sequence to see if you have an indel, an insertion or a deletion using your guide RNA, which is what all of this is showing down here. There are insertions, in this case mainly deletions, these little gaps that you see here, and then down here an insertion. So their CRISPR worked. Any questions on this before we go on? Every single CRISPR-Cas9 paper that we looked at, every one of them demonstrated this, by the way, in different ways. Anything else? Yes.
Do you also technically, like, it wouldn't be very accurate, but like PCR, I know that some papers did that and then they sequenced it.
Yep, so whatever it is, if you have to amplify it before you sequence it, it's really what you're looking for that get a slide 16 at all. Do you have indels in the genetic sequence of the target gene that you have looked at? So the other thing is, this may or may not ring a bell for you. Why do we use a cache domain? Yes.
It targeted the other nuclear membrane.
Perfect. Outer nuclear membrane. I want to see if something is getting, or I want to target something to the nuclear membrane. I might have a cache domain. Perfect. What is the function of the MECP2 promoter? What do we use it for? Where are we trying to deliver our genes to? Yes.
Brain.
Sure. Brain. Brain has lots of things. It has glia, it has neurons, it has neurons. Okay, so this is used to target our gene toward neurons. So keep that in mind. Is that okay for everyone so far? Any questions? Anything you want cleared up? Anything that you want to double check about what you need to know for your midterm? Honestly, it's going to be a really straightforward midterm. I really just want to know, do you know the techniques and how they kind of work? What sorts of things you would look for? So here's an example of a labeling diagram. So you'll have 2 types. One, you draw something, and the other one, you just go ahead and label it. the drawing part, because some of you are not really great artists, no offense. It'll be really simple, so you don't have to worry about it. But here are two separate vectors that were actually taken from papers that we went through in Fang Zhang's lab. So what promoter could he use to target the nuclease in A to all neurons? Well, yes, we could use MECP2 right here. How would he help his guide RNAs target toward the nucleus, and how would he visualize this based on what you see up here? Again, really straightforward answers, nothing tricky. This is what your short answers are going to look like. So tell me which I should be looking for the guide RNAs. Really simple, really simple. A or B? Should I be looking at this, or should I be looking at this? You can't do this on the test, and I have no idea what you're pointing at. So A or B? So we're looking at B. So how would he help his guide RNAs get to the nucleus? What is he using to drive them toward the nucleus? Yes. So we're using this cache domain, and again, you may be asked, why do we use the cache domain? Because it targets things to the outer nuclear membrane. And in this particular diagram, and again, this is a really simple example, but this is the type of labeling diagram that I'm asking you to be familiar with. How is he going to visualize, just by reading this, how is he going to know that his guide RNAs are in the nucleus? Yeah. Okay, so the GFP. So how is he going to visualize? And it's not a question up here, but we talked about it. How is he going to visualize whether or not the Cas9 is in the cell? What might he use based on what you see up here in A? Yes. Right, so we talked about hemagglutinin, or the HA, and that is a way that we can go back in and do it. And those are the types of questions, because you might have slightly different sequences. You might have sequences that you've never seen before, but you should be able to interpret. As long as the proteins are the ones that we've covered in class, or are things that we have talked about, you won't find it really challenging at all. Do you have a question at the back? Sorry, you're going to have to speak up just a little bit. Actually, you should tell me, what is normally we had done this with two different vectors. This is one for MECP2 as the promoter, and then this one, actually, you're right, it uses synapsin 1. What is synapsin targeting? Do you remember? Can anyone help your colleague? What do we use synapsin? It's a promoter for what? So it's also a neuron-specific promoter. So in theory, you could have also said synapsin 1 over there as well. So this is what I mean by being a little bit like a graded potential. There's like a little bit of flexibility, but if it's like for all neurons, like you would say either synapsin, MECP2, or things that we talked about in class. Does that answer your question? You're good to go for next Tuesday? Okay, good. Okay. So other things, other ways to think about this. So what gene is normally a syndrome? So this is how I would ask a multiple choice test question. Yes, MECP2. So lecture 2 is really about MECP2. How much you detect that not in a cell called MECP2, if I knock it out using CRISPR, actually has an effect. And I could look at the density of synapses. Sure, that's one way. What other thing could we do? Yes? Could, theoretically, I would take that answer. Sure, you could do a Western to see what, though? What would I look for?
You would look to see, compared to a control cell, if there was less MECP2 protein.
Okay, so MECP2, great. Sure, I would also buy that. So think a little bit about these. You'll get something very, very similar on your really short, brief answer questions. They shouldn't take you more than 2 or 3 minutes to be able to answer. And these are the style of short answer questions. It's not those long ones that we used to have before. Jenna, did you have a question?
Yeah, would it be like a knockout or would it just be a knockdown effect? Do you
Um,
completely get rid of it?
you would not be able to get completely rid of it throughout the entire brain, for example. It wouldn't be a complete knockout. It would be a knockout in a particular area. Do you remember what area, for example, they did this in? Because that's important. That is a detail that I mentioned. Yeah.
Was it the dentate gyrus?
So this is the dentate gyrus, which gets back into the behavioral assays, which we went through really briefly. No need to describe them. What behavioral assays might you use to go back and test that MECP2 knockdown altered learning? How about someone else? Kai.
What was it that scared association model?
So I have no idea what scared association models mean. For me, that might mean students showing up to the University of Toronto on midterm day. That's a scared association model. So if I have to guess, it's not going to work for you. Anyone else? Yes? Anne? Sorry? So the contextual fear conditioning model is one that we use to test for altered learning, and it's specific to the dentate gyrus, at least according to Fong Zhang. So other things that are in the notes, I'll ask you to go back and review. So this is an example of a slightly longer question, very similar to what you'll get. I don't think most of you will have problems with it. I just want to know, have you looked over your notes and did you understand your notes through either diagrams, multiple choice, or short answer? So what is a DNMT? Do you guys remember what DNMTs are? DNA methyltransferases, and these DNA methyltransferases are expressed really highly in the dentate gyrus. So this is just yes or no. How would they do if you knock down DNMTs in the dentate gyrus? How would they do on an elevated plus maze? They'd be okay, altered behavior, what happens? Any ideas? Guesses? Chloe, could you take a guess? They'd be okay, right? So there's no effect on things like anxiety, which is what an elevated plus maze is taking. Do you remember how they did on a novel object recognition test, which is for spatial memory? Did they do okay? Yep. Yep, they were okay there. And the only thing that was affected was contextual fear. So as long as you sort of recalled all of those details, those are the types of things that on a slightly longer answer I'd expect you to know. I'm not trying to make this a really easy exam, but I just want it to be a fair exam where we're testing for content and your understanding of different things, okay? So there's no need to memorize this type of diagram here. You're not going to be asked to draw this thing out. Just the key features of what dystrophin actually looks like. And we didn't go through all of the different things that are found in dystrophin, but you should pay more attention to part A. However, do you guys remember why we talked about this diagram, or in what context we talked about this diagram in B and C? Yes.
It was because some of them, when they're spliced, the exons have to go beside each other, but there's some that are less important regions, and so then we look at exons.
So this was related to exon skipping. And so if we, for example, decided to knock out exon 24 over here, what's that, or 26 over here, would the two adjoining regions still make a functional protein? How many of you say yes? Okay. How about if we knocked out this particular exon over here, 20, and the two exons right beside it had to repair through homologous directed or homology directed repair, would that still result in a functional protein? So that one wouldn't. So knowing, for example, how this diagram gives us an indication of how exon skipping, being able to bypass one exon so that the next exons beside it are still able to form a functional protein, that might be something that if you're not comfortable with it now, come and see me. It's something that I want you to be able to take away from this course, and it's something that you should expect on the course. And it's, again, it's really easy if you need a little bit of extra help. So other things that I'll likely ask you on a multiple choice, because I like the histopathology on multiple choice test questions. Do you remember what some of the hallmark features associated with tissue damage and muscular dystrophy are? There were like 2 slides where it was like staining. Some were Nissl staining. Sorry, some were H&E stains and some were fluorescent stains. You should make sure you go back and examine those. How do you actually test for a functional? So we talked about behavior in the brain. How about in muscle? How do you know if your muscle is actually getting better? Yep. So we could look at grip strength. And again, you'll get about 3 multiple choice test questions, primarily related to understanding the histopathologic changes. And again, really brief short answer questions from this section. Like literally they're 1 or 2 answer type, okay, like I've been showing you. It's not these long half paragraphs or anything else. Any questions? No? Okay, so from week 3, lecture 3, I have one really easy, and I'll just tell you this right up front, one really easy multiple choice test question from slide 9. So go back and study slide 9. Slide 20 to 21, again, there's a multiple choice test question. It's straightforward, but it's more of an application type that I showed you before. So make sure that you're studying slides 20 to 21. I think this was your question, Jeff, about the SSFO and the optogenetics.
Sort of.
Okay. I'm not really sure what sort of means either, but this will be part of a short answer question. And if you go back and study the slide that's related to the new variant of optogenetics or channel rhodopsins, you'll understand what I mean by that, and you'll probably be able to figure out why it's an important thing to go back and look at. There's no need to memorize this slide, which is slide 24, which just gave you an overview of how we use optogenetics in a number of different settings. You don't have to know a single detail from that slide. Don't bother. Okay, it's just an overview slide. Please don't start panicking from that. And from slide 27, you're likely going to get multiple choice. And again, make sure that you know the Cree-Lox system as well. Any questions about this before I go on? Okay, please don't memorize any of the statistics related to the epidemiology of epilepsy. Here is a really good hint for you. You can start talking about it online. It's more important that you get a good answer rather than anything else. Do you remember what tiagabine is? Yes?
It inhibits the GABA. Like I remember the diagram. So it was the synapse, and then you had the GABA receptor, which inhibits neuron
Yep.
shooting, and the tiagabine inhibits the GABA from going back in.
Okay.
Is that right? No.
I'm not even sure how I'd mark that.
Well, I would describe it better, obviously.
Okay.
Do you also, a question,
Yes.
Would you take a drawing of that?
Would I take a drawing of that?
Yes.
What's
Like a schematic drawing.
So let me draw what you've just described to me. So here is, I think what you're saying is this,
Yeah.
and then you're going to do stuff like this, and you're going to do like GABA receptor, and then you're going to do something like this, and you're going to talk about like tiagabine or something, right? Is that what you're thinking?
Yeah.
No. No. Unless you actually labeled it. So what is this? Like, I have no idea if you actually know what that means. Is that just sort of a vesicle that?
Oh no, I would label it. I would label it.
And what would you label it as?
GAP1.
So this would be that GABA transporter, right? So it inhibits the GABA transporter. So you might want to think, because this will probably be part of your longer answer question, or very similar to your slightly longer answer question. How you might use CRISPR to do the same things like tiagabine does in a model of epilepsy. Okay, so think a little bit about that. Not very difficult, to be quite honest. If you know what tiagabine does, you know how it works in epilepsy, then you might have no issues at all. And again, it's just to see if you understood the material. if you actually add up all of the little things I've been telling you, you'll be able to work out the 25 multiple choice test questions. So this is the FNIRS. You get one multiple choice test question, really straightforward. I went through it really quickly, but it's a really important technique as well. Slide 24 for detail. Why do we actually use parvalbumin? What is CFOS? If you know those already, why we stain for CFOS, you'll be fine for the midterm. But I'm hoping that you're getting the idea that this is a really straightforward midterm. I'm not putting in any tricks, nothing really hard in terms of application. It's really just to make sure that you understand the techniques that we have used. So you get about 4 multiple choice test questions just on lecture 4. I can't remember 4 or 5, but it's around 4-ish. Don't worry about the physiology of the eye, which is the retina, which is what it's saying here. No need to worry about reviewing the physiology, which means the physiology of the retina. You don't have to memorize the retinal amacrine cells, the bipolar cells. You don't have to memorize the ganglion cells. As sad as that makes me, how do you diagnose this X-linked retinitis pigmentosa? Again, going back to mouse models, you'll have to have a short answer question related to some kind of mouse model. Make sure you know those. How to generate these iPSCs step, but not all the details on this slide. Right, takes is to introduce the 4 factors. The only thing you need to know is the OCT, SOX, CMYC, and the KLF4, generating the iPSCs, and then how we stain for tissue that we think is pluripotent, as well as one other pluripotent test. What other one did I mention that you need to know? So the teratoma assay, which looks for what types of tissue? Yep, so 3 types: ecto, meso, and then endoderm. And then there were specific examples that we covered in class that relates to ectoderm, meso, and endoderm. And that's it. If you were able to follow everything on this slide, or that when you go back and you review it on the video and you understood everything, you should be able to walk out of there without too much difficulty and get a 90. I'm not looking to make this a challenging test. Your final exam will be the same format, it's just a little bit longer. There's a little bit more material because we have 4 lectures here and we have 6.5 on the final exam, but it'll be exactly the same format: application, short answer, diagrams. Any questions? I'm available all of next week as well, except for Family Day. You can come in and see me. Do you guys want to do anything online or no? Like a review session online? Where you guys are okay? How about we do this? I will put some extra practice questions for you online. Feel free to discuss them wherever, and then the day before the midterm, I'll post some answers and you can go back and check. Is that okay as well? Okay. I'm hoping you guys do really well, and if you need to ask me about any of the things that you weren't comfortable with here, come and see me at any time over the next 1.5 to 2 weeks. But I do expect you to do like really, really well on it, because we really want you to learn the material and be comfortable with it. Okay, thanks. I'll see you, I guess, in about 1.5 weeks, unless you drop in for office hours. Arsalan, you should write the test.